Okay, chapter 24 is on the immune system. Alright, so the immune system is important for many reasons, and you can see what happens when your immune system is attacked if you look at AIDS. What happens with AIDS is the immune system becomes under attack. It is called acquired immune deficiency syndrome, and it's an epidemic. You have thousands of people being infected every day by AIDS. All right, HIV, human immunovirus, is the virus that causes AIDS, and it attacks the immune system, and it destroys the body's ability to fight infection. So it's a nice example of what would happen if your immune system was under attack. There's also cancers that attack the immune system, and any little infection becomes a big problem because your immune system isn't working properly. Okay, there are different levels of immune system defense. So the first basic level is your innate defenses, and that includes things like the skin, not having your blood and stuff directly exposed to bacteria and viruses, but to have a protective layer. You have mucous membranes that produce mucus, and that's actually really important in your nose. And you have all this snot and stuff, and we tend to just think of it as annoying, but it actually has a purpose. When particles get in there, they get trapped and then ingested, and they don't end up in the blood system. It protects your mucous membrane because your mucous membrane is a point of entry for things like viruses and bacteria. That's why if you touch a door handle that's all nasty, full of bacteria and viruses, and then say rub your eyeball, that membrane is a point of entry. You have like macrophages that are phagocytic cells, and that means they eat anything, right? Phagocytosis, remember, is cell eating. They're going to eat any invader. And you have antimicrobial proteins, actually, that can shut down viruses and bacteria. So you have innate immunity, and this is present before you're exposed to a certain pathogen. This is just always there. Okay, when something like a bacteria breaches the body's external defenses and gets into your body, we have something called a macrophage, and this is going to go around and it's going to engulf and eat the bacteria, and you cannot tell me that picture isn't awesome. Look at that beast. That is a cell that can catch bacteria and engulf them. Crazy cool. All right, you have something called interferons. These are the proteins I was talking about that are produced by virus-infected cells, and they help other cells resist the virus. So when a cell has been infected, on the left side over here, you have a cell that's infected over here. It turns on this interferon gene that's going to produce the proteins that's going to stimulate other cells to produce the protein that's going to block the virus. So it's kind of a cool little process there, and those are interferons produced by an infected cell. It's going to go out and then go to another cell and then hopefully kill that virus. All right, so inflammation is another immune response, and we talk about inflammation nowadays as if, oh, it's such a bad thing, but we're talking about chronic inflammation when somebody says it's bad, or eating these, you know, non-inflammatory foods or whatever. That's chronic inflammation. But basic inflammation, when you have had damage, is actually a good thing. It allows, so let's say you have a cut over here, you get a cut, and you end up with bacteria. All right, so the first thing you do is you have chemical signals. You are going to have probably histamines that are being released that are going to cause inflammation. So if you take an antihistamine, it will reduce inflammation, but in this case we want it. It causes the dilation of this blood vessel, and when that happens, it becomes leaky, which is good because these white blood cells that would be trapped here now can kind of ooze out between these epithelial cells out into the interstitial fluid and go get and kill and literally just engulf. Phagocytes, They eat the bacteria, and so that inflammation is actually a good thing. Okay, the inflammatory response can disinfect tissues and limit further infection. So hopefully they get into the tissue and it doesn't make, the bacteria don't make it into your bloodstream. It just all takes place very locally and it gets destroyed. Okay, the lymphatic system is crucial for your immune system as well. And you're going to actually have your white blood cells in your lymphatic system. So you'll have your B and T cells, and they will go into this fluid here, this lymphatic fluid. And I'm sure you've heard of lymph nodes. You have some up here. You have a bunch under your armpit. They can become swollen when you have an infection. But one thing they do is they have all these white blood cells that are available to go attack. They're available to go attack things in your body. And if you look off to the right-hand side, you can actually see this lymphatic capillary is just as small as your regular blood capillaries. And this is your interstitial fluid here. And this lymphatic capillary, it can take extra fluid into it. So if you've had a bunch of lymph nodes removed, because you've been diagnosed with cancer, you'll actually have trouble getting rid of fluid in your body. It's one important job these lymph nodes have. Okay, the vessels collect fluid from body tissues and return it as lymph, and that's the fluid, to the blood. The lymph organs, such as the spleen and lymph nodes, are packed with white blood cells that fight infections. So your lymphatic system is actually quite important with your fluids in your body, as well as housing those white blood cells. All right, your immune response counters specific invaders. So our immune system responds to foreign molecules called antigens. And antigens are, it's a shortened word from antibody generators. So anything that generates antibodies in your body is called an antigen. And the immune system reacts to antigens, and then it also remembers it, so that if you get an infection, hopefully the second time you're exposed, your body's faster at fighting the infection. Okay, we can temporarily acquire passive immunity by receiving pre-made antibodies. And passive immunity means that we are not making it ourselves. Babies can have passive immunity from their mother's milk. But anytime we talk about our own bodies making antibodies, it's called active immunity, whether it is because you caught, say, the chicken pox, or whether it is because you got the chicken pox vaccine. Doesn't matter. Those will both be called active immunity. All right, your lymphocytes mount a dual defense. All right, so lymphocytes are a white blood cell. And you have two types. You have B cells, and think B for bone marrow, and they attack antigens. And you have T cells, think T for thymus. They both start in the bone marrow, but actually the T cells end up in the thymus. They both have antigen receptors. So you see those purple flags outside of the cell. These are antigen receptors. This is what recognizes the invading force, the non-body, the non-self is what it's called, some foreign invader. And these are both in your lymph nodes, your spleen, and so they are ready to fight. Okay. There are millions of kinds of B cells and T cells, each with different membrane receptors. And they wait in the lymphatic system where they may respond to invaders. Well, what does that mean? Each individual cell only fights one specific antigen. It only is going to bind to one. So let's go back to our chicken pox example. There was one cell initially that was had the little flags on the outside, the little antibody flags that are going to recognize chicken pox. And what that one cell does is it clones itself over and over and over again if you get exposed to either the vaccine or to the actual disease. But each B cell and T cell only recognizes one thing, and it waits. And so there's, millions of things that can invade our body potentially. There's all kinds of bacteria. There's stuff probably that we are protected against that doesn't even exist. We literally have millions of kinds of B and T cells. Each individual only protects against one thing. But we have millions of them, so hopefully we're protected from everything. Okay, the antigens have a specific region where the antibodies bind to them. Remember, an antigen is anything that will generate an antibody. And there are specific regions where they bind. So here you have your antigen molecule, and then you're going to have the antibody. So here's your antigen molecule, and you're going to have your antibody molecules that can bind to that antigen. Okay, so when we talk about your antibodies, when you have an antigen that enters the body, it's going to activate a small subset of lymphocytes with the complementary receptors. So you saw on the previous slide that antigen bound to 3 antibodies. Well, sometimes that's the case. Sometimes it only binds to 1. How do we attack it? Well, the specific ones that it binds to are going to reproduce like mad and go out throughout your body and attack that antigen. And so they clone themselves. Basically, they're cloning themselves. That's why you call it clonal selection. It's going to be an army of those, the one that binds to the invading antigen. Okay, the selected lymphocytes multiply into clones of short-lived effector cells. These are the ones that are going to go out in mass and protect your body, find all the antigens. They are specialized for defending against that antigen that triggered the original response. Okay. The steps of clonal selection, you have a primary immune response. So you have some invading force. It's going to produce effector cells, and it's going to produce something called memory cells. So the memory cells aren't going to fight it this time. They're just there to remember in the future if this same antigen reintroduced into your body, the same antigen, your body will be able to attack it faster, and that's because of the memory cells. All right, in the secondary immune response, the memory cells now remember it. They are activated by a second exposure to the same antigen, which initiates a faster and more massive response. That's why sometimes if you have a cold, you can't catch that same cold again. Now, colds mutate very, very rapidly. So if it goes around, the entire class and comes back to you, you can sometimes catch a second version, but you usually won't get sick from the same cold you just had. Okay. So here you go, primary and secondary immune response. So start up here. You have the antigen that is bound to this antibody. This is the cell. It is going to create 2 different kinds of cells. Okay? It is going to create these effector cells, and all these guys do is they pump out antibodies. And if you look at the amount of rough ER they have, they are loaded with rough ER. Why? Because that is necessary to pump out antibodies. It is something like 2,000 antibodies a second per cell. Tell me that's not amazing. So these things are going to pump out proteins all day long. These are going to go bind to the antigen, which is going to flag them for destruction. Then you have the memory cells over here. These are going to hang out in your body just in case this thing invades again. So these just hang out. Now let's say, sheer dumb luck, you get the same cold twice. It invades again. Now you have a ton of these, and they can clone out a bunch of these effector cells that will kick out tons of antibodies very, very rapidly. Okay, so here you have primary response versus secondary immune response. Your primary immune response is always slower. So you have your primary immune response here. So here you have antibody concentration, right? So the more antibodies you have, the better you are to fight whatever this antigen is. So here you have, one sitting in your body. You get exposure to this antigen, whatever it is. Your body fights it. It takes a while. You feel kind of crappy for a couple weeks. Then you get exposure to the antigen again, and your body is ready this time. Look at it. It's going to shoot up the number of antibodies and just pump them out. This graph is also the reason that people who are allergic to peanuts have a far worse response the second time. It's usually not the first time they eat peanuts that can kill them. It's the second time. And the reason is just what you see on this graph. That second immune response is much more severe than the first. All right. Antibodies are the weapons of humoral immunity. And remember, humoral just means fluids. It's from the Middle Ages, when they thought everything was a humor, were all the fluids. And we won't go into a history lesson with that one. But humoral immunity just means in your fluids. So antibody molecules are secreted by plasma, the effector B cells. So here is a more in-depth look at what you were seeing in the other slides. Just look like little Ys, little purple Ys were your antibodies. Okay. Here it is in more detail. You have an antigen binding site. So here you have the antigen binding site that's going to bind to the antigen. And then you have the heavy chain down here. The heavy chain is what actually flags this thing for destruction. All right. So antibody marks antigens for elimination. Antibodies promote antigen elimination through several mechanisms. So how are they going to do that? One, you can physically, so here we have the production of these antibodies. It can just block the virus from binding to something. It can block the bacteria from being able to get to your cells, just physical blockage. Agglutination is just clumping them together so that they can't do what they need to do. Precipitation is very similar to agglutination, except that it's going to take them from a liquid form to a solid form, but it clumps them together so they cannot invade. All three of these cause these things to get eaten by a macrophage. So the phagocytosis here, they're going to come in and destroy it and eat it. These three things will cause this to occur. There's another type which actually activates this complementary system. And this is the foreign cell. So this is not your body cell. It's a foreign cell. These will bind to the cell, and it's actually going to lead to lysis. It's going to cause the cell to explode. So that's another way they can do that. They'll actually poke holes. It'll explode. Okay, you can make monoclonal antibodies if you want in the lab, and they can be used for different things, for diagnosis and the other kind of things. And the way you do that is you're going to fuse two different cells. So this is in the lab, so they're going to have a tumor cell, and you think, ha, tumor cell, bad. In this case, tumor cell, good, because the thing with cancer cells is they divide indefinitely, and since we're leaving this in the lab, we want something to divide indefinitely. We don't want it to live for a couple weeks. These will live indefinitely. So we're going to have the cells that live indefinitely, and then we're going to take an antigen, inject it into the mouse. We are going to take the specific B cells to this antigen, and we're going to fuse these cells, literally fuse them to a hybrid cell. Now in the lab, we have a colony of cells that's going to do nothing but kick out the antibodies we want, and it's going to be one specific antibody. It's going to be a clone, a single clone from that cell. So you're going to take one cell, and it's cloned. So you're going to get repeated use of one antibody. If you just want one antibody, that's how you get it. Okay, so those antibody molecules are useful in research, diagnosis, and treatment of certain cancers. So they can be used for that. They're the ones when you do pregnancy tests. That is also a monoclonal antibody. So they're used for different things. Okay, helper T cells stimulate humoral and cell-mediated immunity, and kind of like their name suggests, they help out the other cells. And then you have something called cytotoxic T cells. Those are the ones that just go around killing things. They're pretty awesome. I obviously like the cytotoxic T cells. These are the main effector cells of cell-mediated immunity. Helper T cells also will stimulate humoral responses. Okay, in cell-mediated immunity and antigen, you have an antigen-presenting cell, and it displays a foreign antigen on one of the body's own self-proteins to a helper T cell, and it is much easier to understand in a diagram. Okay, the helper T cells receptors recognize the self-non-self complex, and interaction activates the helper T cell. The helper T cells can activate cytotoxic T cells and B cells. So again, they help activate stuff. That's what they do. Okay, and I obviously like all that in a happy diagram is much easier to understand. So, here we go. Over to this lovely macrophage. Start with, here's a microbe, bad, we eat it, yum yum. We chop it up into little pieces, and we have these little self-proteins here. These little self-proteins are going to grab the bit of microbe protein, and they're going to pop it out and display it on the outside of this cell, and I think this is so cool. It's flagging this thing, like, look at this. This is how we identify an invader. So it puts these little flags out all over the cell, the outside of the cell, and this is called an antigen-presenting cell. It's much easier to see this visually here. The helper T cells binds to this, and what it has to do is it has to bind to the self-protein. This is your own body's protein. The green one is you. And then this little orange one is the invader. And the helper T will recognize, oh, look, I need to, like a lock and key, really. Like, here it is. I need to look at this invader. And that is going to activate this thing. It is going to produce this interleukin-2, which is going to stimulate cell division, both of more helper T cells. So it's going to make more of itself. It's going to start making more helper T cells. It is going to stimulate division of the B cells with the same flag, all right? So that's your humoral immunity, where I said it in the previous slide. I said it also helps with humoral immunity. This is why. It's going to spit out more B cells. If you recall, they are going to produce tons of antibodies. It also produces the cytotoxic T cells, which literally attack and kill infected cells. These are the cool ones. So that's how that works, and this is the antigen right here. This one's the antigen-presenting cell. So a really cool way of identifying stuff. All right, why HIV is so bad? It destroys those helper T cells. So again, it's the good example of what happens if you have a destroyed immune system. And I just mean good example, like it's not good, but it's an example of how important your immune system is. Okay, cytotoxic T cells destroy infected body cells. So these things are cool. What they do is they have been activated. They recognize cells that are infected. So you have an infected cell, and it's displaying this self-non-self, and this thing goes, this cell is infected and producing virus, bacteria, whatever, not doing what it's supposed to be doing. So it releases something called perforin. Think perforated here. It's going to perforate the cell membrane, but it's a protein called perforin. It's going to come over here, and it's going to cause this thing to destroy itself, hopefully stopping the infection. Okay. Cytotoxic T cells may help prevent cancer. In fact, a study just came out a couple of weeks ago, so January 2014 or so, that really showed that cytotoxic T cells actively kill cancer cells every couple of weeks. That without your cytotoxic T cells, a lymphoma would develop within 2 or 3 weeks in an average person. So that tells you every couple of weeks they go through your body and kill off cancerous cells. Okay, the immune system depends on our molecular fingerprints. So it only reacts against non-self substances, not against self. This is why it can reject transplant organs, because the organ is going to have the fingerprint, the proteins, these little flags are going to be of somebody else. So the more they know about transplants, they now test tons of different proteins. They used to just test like, okay, you're A or type O blood, and a few proteins. Now, before they do a transplant, they're looking at least 20 different markers to try to get the self, non-self reaction minimized. And that is called graft versus host disease. The host is the person that it's going into, and the graft is the person, they've grafted an organ into this person, and it can cause the immune system to permanently attack the new organ if it is not close enough. Okay? Things that are bad for your immune system. So there are all kinds of autoimmune diseases, all kinds of them. Lupus, they think things like ulcerative colitis are autoimmune. There's tons and tons of them. Anytime the body attacks its own self, it is sort of an autoimmune disease. It turns its attack mechanism onto its own molecules. Then there's immunodeficiency diseases, and that's when you have immune components lacking, and then you get infections, whether that's from having HIV or cancers can cause that. And then there's physical and emotional stress, and that can weaken the immune system. Probably your grandmother would say, you know, don't go outside in the cold, you might catch a cold. Well, where did that idea come from? Obviously, you know, you don't catch cold, you're getting a virus. Those viruses are around you at all times. We are surrounded by viruses and bacteria, and our body usually defends against that. But when you are physically or emotionally stressed, your body doesn't defend as well, and that's when you get sick. Okay, allergies are overreactions to certain environmental antigens. So it's an abnormally sensitive to an antigen or an allergen in the surrounding. The one I gave earlier was the peanut one because that can kill you. But there's all kinds of allergies to all kinds of little things. You know, the proteins in pollens can cause an allergic reaction. It's an overreaction to normal antigens in the environment. Okay, so you have two stages. This should look very similar to your primary and secondary exposure, where you have antibodies, but here we're also looking at histamine being reacted. Remember histamine, released. Histamine's going to be released. Remember we talked about histamine being part of the inflammatory response, which can be good when you need inflammation. However, in the case of allergies, we are going to release this is histamine inside the cell, right? So we're going to release that histamine when it's exposed to that antigen, the second exposure, and it's going to cause swelling when you don't want swelling.